Hello.
Hi, Wendy. Can you hear me?
Hi. Who's going to take minutes today? Who's going to be hung? I just checked the MycetOS site. I think Erasmus MC is the one who's going to
Yes.
convene, and somebody else is taking the minutes. Is that correct?
Yes, so Erasmus is going to be the host, and taking minutes is going to be someone from UCL.
Okay. So I see that Matt and Kimberly also came in. Mickey and Ma
Hi, sorry,
are there.
yes, I was just delayed. What were you sorry, what were you saying about the roles and so on? Sorry. Responsibilities,
So yeah, I just checked with Hang, and on MycetOS, I think, well, we were supposed to do the minutes last time, but I was a little bit late. So Kimberly took the minutes. Thank you for that, Kimberly. So I think that now we should host, and somebody, either you or Dimitri, should take minutes.
right.
It looks like, well, usually the one who takes minutes rotates into the host of the next meeting, and then the third lab is taking the minutes.
Yeah. Can you also create a new GitHub issue for this month meeting after the meeting as well?
Yeah.
For the January.
Was the last one November? I guess it was, right?
So
Okay.
the minute taker or the one who's responsible for minute taking should put a new GitHub issue for like this one. That was
So
it.
the
Okay, I will put a new one in.
Yep.
So Matt, is Dimitri also joining, or do
I
you know?
thought so. Yeah, it should be. Hopefully.
Okay, yeah,
Are
maybe
we-
due to the so maybe due to the sake of time, let's start. Are there any pressing things? Maybe the first agenda item should be the time in which we host this meeting, because there was a lot of debate on GitHub. I think for me, this time is okay, but I would also be okay moving it up to 10 o'clock. I think for Matt, it's probably the same, because for us, it's the easiest time, of course, because for us, it's broad daylight, but I can imagine that for Kimberly and Hang, this time is not very convenient. So what would your suggestion be? Move it to 10 or do the rotating schedule again so that the United States can also join at some point, because for them it's 4 o'clock in the morning. On the other hand, they have not always been around, and you have. So
I think that's my thing. Not knowing if they're going to be there each time, and then us staying up late, and then unfortunately no one makes it. But I think doing the rotating thing's the best way to try and get around that and get to a good compromise for everyone. So having a slot and a B slot, and each month we rotate between them.
yeah, because when is because the next meeting is on February, so then I would suggest to take that one at 10 o'clock our time. So that's the time you proposed.
Yes,
But when is the time change again for you? Because maybe that's
April.
also
April.
Yeah, early April.
April.
Yeah, so basically, can we just one of the rotation will be 2 hours before like this time essentially.
Yeah.
Right.
Yeah, so then 10 o'clock, 12 o'clock, rotating like that for the upcoming meeting. So we will set the meeting for February at our 10 o'clock. So that's 2 hours earlier than this meeting. 3 hours earlier as this meeting, I think.
Yeah, that's 10 EU time, right?
Yeah.
Right.
And
That's
then
fine.
the other one we leave at this time point. And then we will look when the summertime, our summertime is here and your winter time, when we move the time again. So maybe that's the best way to go forward, and we put it on the agenda for next time as well. Because then we will have a clearer view of the summertime maybe. So everybody can think a little bit on it.
Sounds good.
Yeah, the time change makes it awkward, right? And just because we can't sort of set all the meeting times now. Is that that's the issue, isn't it?
I think that last time Hong made a suggestion that we keep the same times, because I think we change and then a month later you change. And so then once we've both changed, it's okay again, because it's back to the same time. But it's just that little bit in between where only one of us has changed and the other hasn't. And that happens twice. So it feels like a lot.
But if we're fourth,
yeah, I think for us it's March.
Right. If we're
I
like
think
the
the
fourth
UK and
Tuesday or something.
Europe.
Sorry, carry on, Wendy.
Sorry?
Carry on, Wendy. Sorry, that we were talking at the same time.
Yeah, so I said I think that the UK and Europe still change at the same date. So for us it's the same, but I think it's different from Australia. And if I recall correctly, for us it's the end of March.
So ours
when
is
we
the
change.
yeah. So ours is the 3rd of April. So there's a chance that the last meeting we have in March might be just before yours changes, and then the one in April will both be changed. So it might not be an issue. But
Yeah. Right. So as it stands, then so February and then alternate months is the 10 o'clock EU time, and the other ones are 1 o'clock EU time. Is that the idea?
I think so. Yes.
Yeah, and for us, I just checked the summertime ends at the 27th of March, starts at 27th of March. So it's only 1 week difference.
Mm-hmm.
So it will not intervene because we will change directly after this. Well, we have the meeting the week before we change. And if you change the first week of April, then you will have been changed as well before the next meeting in April.
Yeah.
Right. And I think if we just have a agreed final email list of everyone who needs to be invited, then it's easy to send out Zoom invites, which will go into diaries. Yeah. We just need to make sure that that's got everybody on it. I'm not sure we have that, right? Unless someone's got an email list.
Yeah, because I just created the ICS files last time so people could just download them and add them to their own calendar. But we do have one set up that Hung, myself, Alice, and Peter are all on, so we can always add other people to that one.
Is it an email list?
Oh, we just have a meeting that's in for every month set for the Tuesday and the time that it is for us, just so that it's in our calendar and we don't forget.
Right.
But yeah, so I could add other people to that. And then it should work with time zones through Outlook. As long as everyone uses Outlook, that's the thing. Because I think some people use Google Calendar as well. So last time I created ICS files for Outlook, and I created whatever the other type is for the Apple calendars in there.
Right. I mean, that would be convenient, yeah, if you could do it through Outlook like that, because that always works for me, and it always takes into account the time zone.
Mm-hmm.
Yeah, and I also use Outlook, and I think Ma and Mickey as well. I think maybe Wilson was the one not using Outlook, but he will not join many meetings anymore since he has a different job. So, yeah, I think Outlook will be great.
Okay.
Then for the next issue, I think that was the chemistry. I see that Hang and Dimitri are both here already. Well, maybe either Hang or Dimitri would like to update us on the SAR.
Yeah, sure.
Yep. Yes.
Hello, everyone. Hang, do you want to go first, or should I?
Yeah, sure. So I have done the. Let me just quickly share my screen. Okay, so I put my SAR writing, my analysis for the SAR onto the Google Doc that we share on the GitHub page. So where I think Wendy and Wilson has put the link before, so I just put my writing there. So basically, I tried to put it together into like a nice narrative. So currently it's about 6.5 pages long. So obviously we need to shorten that up with subsequent edits. But I put together all of the thing all of the patterns and analysis and discussion that I've seen by just looking through the data. So I was still, I asked Dimitri earlier about the match pair list for the second motif as well, and I can once you're done with that, Dimitri, I can have a look at that and if there's anything that's interesting, I can put that into here as well. But with just what I've done, what we've got so far that I have
Sorry
here.
for interrupting.
Sorry.
Issues?
Sorry. So you got cut off a little bit when.
So I can't hear very well. Explain problem or not?
You're breaking up. Your audio is breaking up a little bit, Wendy. Sorry, I can't quite hear what you're saying.
Okay. Yeah. So basically, I started with the leads that was from our previous publication. So that was these 1, 2, 1, 2, 3, 4, 5, and this is actually a link that we have after the publication. So there's a mix of the previous and the any lead, which I would then just put into generalized sort of like scaffold that I base my analysis on. So of our evaluated compounds will either fall into one of these two groupings. So basically, the base structure that I focus on is the 3 aromatic rings surrounding the cores, and then obviously one of them have the nitrogen ring, so that's make it the pyridine ring, and then the other 2 will have various substitution patterns. And the difference between these 2 scaffolds is basically just on this ring here, where it got changed to a pyrazine ring or related scaffold here as well, with various linkers. So the change between these 2 is just this part here. So then I started to do the analysis on this first one, which is this here, and basically group together all the changes for this. So basically, so ring 1 here, the best scaffold in this part is 3 pyridyl group, and if you change anything else, it's basically it just makes it less potent in vitro. So the color coding was from what we agreed before between the sort of like the traffic light system of like very good, kind of like middling, and then the bad, and then the black one is actually things that I'm still waiting for an update from the IC50 and MIC50 values. I think there's 4 compounds that I still haven't seen the IC value, but that's why I can't. That's why some of them are still black right here. But basically, the core is quite tolerant, but the best option is actually still the OH here because of the in vivo result as well, which will come to later. But basically, there's a good reason for this. Is why I had a look some of the because we were talking about the sort of the probable mode of action of the CYP51 inhibition by azoles as well. So I'll just have a look at some of the crystal structures from the literature. That was for, I think, so we have crystal structures of azole-bound CYP51 for things like tuberculosis, or these 2 fungus for humans, also for the protozoan from Chagas as well, which obviously don't have fun for mycetoma just yet, but I found them from the protein database databank, and then I just look up of the papers that describe the structure. And there are good reasons to why if phenomenal analogs also sort of like follow this mechanism of action, which we don't know yet, but this it sort of like provides evidence to that. Then there are good reasons why some of these motifs are actually active. I'll talk a little bit more about that, but basically the core here, OH is best. If you remove if you just sort of remove the functional group completely, then it just loses its in vitro activity. Therefore, the two remaining two rings, so there's various substitution patterns. So the X on purple here are various substitution on this ring, and then the Y is for the other one. So basically, dye substitution is best, and then if you kind of remove that, then just a single substitution is not that very good, and then if you remove the substitution, then it's also kind of not good as well. And then on the other side, we haven't really had a lot of the meta substitution, so the 3 or the 5 position. That's why I kind of recommended that we sort of like ask my team to have some more of this shipped to Wendy for testing. We don't have a lot of this to make to sort of like know for sure the effect of the meta substitution, but from what I've seen, ortho substitution, so the 2 substitution in this position here are actually quite good. Now, if you don't have any substitution at all, but the other ring has substitution, especially if it's dye substitution, then it's still good. But if you have, instead of a phenol group, you change it to a benzyl, then that would just completely destroy the in vitro activity. So the spacing is actually quite important as well. And that's also consistent to what we're going to see a bit later in the other scaffold as well. Most of the hetero cycles that is on this, that would go to this ring here, not good. So just, and if you have aliphatic rings here, then it's sort of like mixed. But if it goes with the dye substitution on the other ring, then it's good, but otherwise it's kind of just meh. So what I did was I also create a matched pair for this scaffold as well. And the reason that this is good is if you have a look onto, say, one of the structures, say, for example, this one right here, because there's 2 rings right here, you can actually just either look at it in that way, or you can actually just flip it the other way as well. So say, if you flip it the other way, then this ring is going to be go over here, and then this ring is going to be over here. So the matched pairing, when I did this, I actually took into account of both of that configuration. So the normal and then the flipped as well, just to see all of the possible combination with the pyridine ring on top. So basically, if it has, I consider the 2 substitution on either of the rings, and then the 4 substitution on either ring, and then no substitution on either one of those. So which does actually show that with a 2 substitution, is most of these will give really good in vitro activities, and then there's 4 are like the middle, that's sort of like mixed between the 2, and then this is the worst. But the best. Wendy, I think from memory, I still have a few holes in the MIC. So the 175 hasn't have it yet. So I just checked this morning as well. 175 is still missing 2. 171 and 170 are also missing. And so that means I don't know if it actually got evaluated in vivo as well. And there's 174, which has excellent MIC, but I don't know if whether we're just too late for the in vivo, or it's going to be done later, or it's done already, but it hasn't been updated yet. So I'm not sure. I was going to ask you that a bit later as well.
Yes, so I can answer that. For the in vivo, everything is in there which we were able to test. But remember, at the end of the larvae season, we were not able to test everything we were supposed to test because, well, the larvae season ended, so the time ended. And for the IC50s and MIC50s, we updated it last week. And according to our files, all the things which had an activity below 20% at 25 were there. And I saw one of your compounds who had an activity of 38%. That one would not be selected for IC50 or MIC50. So that would not be there. So it's the 070, 0170.
Yeah.
That would not be there. I have to double check 174. Oh, that's there.
So 170
171.
and 175.
Yeah, if Ma, do you know by head if those were tested in the last round?
Actually, I'm not sure now. Let me check later.
Okay.
Yeah, I can't remember that.
Yeah, so once you let me know, I can update that, and I can update these two as well.
Okay.
From what I can see so far, this is the result. So disubstitution on one of the ring is best. If it's not possible, then the 2 substitution is still better than 4 substitution, and without any substitution at all, then it's just not good. And the reason for that is if you have a look at the compound here. So this is based on existing literature evidence of azo-bound CYP51 crystal structures of a few different species. Now, the CYP51, from my understanding, that it's sort of like it varies a little bit between the different kingdoms, but there are parts of it that are conserved, which then sort of like let us do its job between these different organisms. But what I found so far is there is an active pocket of the CYP51 enzyme, and this drug or azo drugs in general will normally be able to fit 2 rings inside the pocket, and as well as this OH here, if it's there. And obviously the pyridine ring is going to coordinate to the ion of the heme, which then blocks the enzyme and inhibit its interaction with this normal substrate, which is the lanestrol. Now, this hydroxy group here and the 2 the disubstituted phenyl group, so the disubstitution here, it actually interact with other residues inside the pocket, and it actually form either so this one definitely will do a hydrogen bonding, but this one also will also interact with other residue, and it actually stabilize that the whole compound, the whole complex, so it's just make it basically stick to the heme a little bit better. So it disrupt the hydrogen bonding network that's normally in there, so it stabilize the target compound, in this case hopefully will be our phenylalanine analog. And then there is final rings here, which usually sit so like further away, so like out in the entry to the active pocket. So some of these, like say this one, for example, would be a little bit longer, so it will stick out a little bit further from the access channel into the active pocket, and that would interact with the other residue at the entry, which further stabilize the drug enzyme interaction. So that's why some of the azo drugs actually have quite a long tail, and all of that is just to stabilize the interaction of the nitrogen that will coordinate to the heme ion of the ion of the hemes. So actually, the essential bits would be the pyridine group here, and then everything, all of the other bits just help stabilize that interaction a bit further by grab onto the different residues that's either in the pocket or at the entry to the pocket. So that's how I understood it anyway. And that's I've read probably 5 or 6 papers that describe this for humans, for tuberculosis, for the 2 fungi, so the albicans and the fumigatus, and also for Chagas, it's also T. cruzi. I tried to search for in the protein data bank what I found is paper, but there's none for mycetoma, so nobody has managed to crystallize one for mycetoma yet, unfortunately. So that will have to be if we were to follow that direction, that will be something I guess we'll have to do in the future.
Yeah,
Is there
so
any-
what we did, Bertrand did make in silico a model based, because we do have the protein sequence, of course.
Yeah.
And he used some models to make a model of the CYP51 enzyme. There are two variants actually in the natural population with one amino acid difference. And for us, it was important to know where that one amino acid difference was. And that's why he did that. We don't see any difference, though, in the natural population between the two variants and their susceptibility towards azoles. That's also because that one amino acid difference is outside the binding pocket. Yeah, but he made those models on the computer. I don't know if it's useful for you to have those, but yeah, we do have those. But
That's
it's of course not a crystal structure.
Yeah, because what I've seen is one of the paper that actually describes, so they were studying the fungus fumigatus. What they do is, I think they were having problem. They also did a computational study as well. So I think they were having problem either crystallizing the structure of the azole with the fumigatus fungus. What they did is they did a homology study. So they actually run a computer simulation of this sequence folding, and then with azole in it, and they superimpose, I think, on a CYP51 azole-bound CYP51 for human, and also a azole-bound CYP51 for tuberculosis. So they did a homology study, and they kind of like found this similarity and differences. So I guess in that sense, it could also might be useful. So just to have the in silico version of the protein. But I'm not that like extensively knowledgeable in biochemistry, so I probably need some help if I receive that, which I'm sure we can get some help if I need it. But yeah, I think that would also be good as well.
Are there any crystal structures anywhere with something phenoramal-like bound?
Not phenoramal in particular, but they have ketoconazole, itraconazole, posaconazole, fluconazole as well. So sort of like in that realm, but not the phenoramal analogs like itself.
Right. And Wendy, could we get hold of some of the protein, or is there a problem in expressing it?
Well, we have expressed proteins from Madurella in the past. Usually we use E. coli for that. But of course, E. coli is a little bit different from Madurella. So you could have differences in folding. We could try, but I never tried it for this one. I tried to express some antigens, and with some of the antigens it worked. But I don't know if the folding would be similar, because yeah, to the extent I express proteins is simply putting a histidine tag on it, take it over a nickel column to purify and see if it's antigenic. So I never did crystal structures or whatsoever with it. And I also, yeah, because we usually use SDS page to see if it's antigenic, that takes out all the folding. Yeah, that experience I don't have.
Because the Seattle Center, the Structural Genomics Center for Infectious Diseases, SSGCID, those guys do, you can ask them to try and do protein expression and crystallization for things to do with infectious diseases, provided everything is openly available. And if they get crystals, if they get something suitable, then they can do thermal shift analysis to see if small molecules are binding the proteins, and if they are, then they can solve the structures. So I can raise that with them to see if we could try this, but I guess the question they would have is what's the likelihood of them being able to get the protein in the first place? That's the only issue.
Yeah, so far I didn't have experienced much problem with expressing any protein from Madurella mycetomatis with an histidine tag on.
Right.
the amino acid sequences and the DNA sequences are there from both variants. And I would suggest them do both because they're equally, well, they're almost 50% present in the population. It's only one amino acid difference. But although it doesn't matter much for the azoles, it doesn't mean it doesn't matter for the phenolimols. And since there are so few fungal structures of CYP51 present at all, I think it would be a good addition. On the other hand, if they were not able to successfully do it with Aspergillus fumigatus, I guess they tried, that might give us a clue that it might be difficult. But yeah, nobody tried for Madurella.
Yeah. So the only other fungus that I saw was the albicans, which was a yeast that was successfully in crystallizing that structure. But yeah, I think they were just having some problem with fumigatus. Yeah, I didn't read. I just went quickly over, so I can't remember what the problem was essentially.
Yeah, so if it was the paper of Evelien Snelders, where you got the structure from Aspergillus, I know Evelien, she's from Nijmegen. They simply didn't have the experience to do a crystallized structure. So they had this collaboration with this bioinformatic department. That's why they did it that way. It's not that it was difficult to get the crystal structure, but they didn't have the experience.
Yeah, it was Evelyn Snelders that was paper in 2012.
Yeah, so that doesn't mean that it's impossible for Aspergillus fumigatus, but I don't think they tried to do the crystal structure. They did this for her PhD. It was one of her projects.
Yeah, and the other structure I found from a fungus is the Candida albicans, which is this one right here. That was a Hargrove paper. But yeah, only two.
Yeah, Candida albicans is of course one of the most common fungal pathogens. So it is also the most likely choice they would tackle because Candida is more common than Aspergillus fumigatus in disease.
Yeah. Yeah, so I'm just going to quickly go through this as well. So not much going over. I've written everything down, so what I'm going to quickly go over is the thing that I found as well. So for the second scaffolds that I found, it's just the same thing again. So the 3-pivotal gives the best in vitro result, and you also see here that dye substitution is best, and then still waiting for this result. I forgot what it was, but yeah, if you remove one, then it's just drop off your in vitro result. If you put a spacing again in the third ring, so any kind of spacing, so this is similar to what you saw up here with this one right here. So if you do that, and it's just kind of is bad. Still, what we have so far is that they are bad, but still waiting confirmation for the two black compound from the IC50 value. And I'm just explaining the different combination that we did here as well for the piperazine, the piperidine, and others as well. And the reason that I did here as well is because we don't actually have an OH group right here, and the reason for that is the OH, the hydroxy group in combination with the amine right here, or with this amine here, just makes the compound unstable. So it's just it's not possible. That's why it's not really there. So it's just a H here. And you remember from before, the H in that position is that OH group actually removed one of the stabilizing hydrogen bonding to one of the residues in the active pocket. So actually, if CYP51 is to be the hypothesized mechanism, then this G2 scaffold is kind of a little bit worse compared to the other one because it's lacking that OH firstly, and if you just remove the dye substitution, it's just getting worse as well. So I think we might have got lucky with sort of like this one here in combination with the piperazine and this linker with this group here, but then we didn't really find anything that sort of like surpassed that in terms of the in vitro and in vivo activities, except for I think two of Dimitri's compound, which is I think this one right here and this one. But then I think only this one has a good in vivo. I haven't kind of gone up to that yet, but my reasoning from what I've read so far is that, yeah, just because we're just lacking some of those further stabilizing effects. However, I think that because I think the in vivo angle with the log-D values, like the lower log-D value, might give a higher in vivo test. I think that angle is still like promising from what I've seen so far. But I think there's still like two layers of that as well. So you still, because if this is to be the CYP51 is to be the mode of action, then you still need the components, which is the pyridine ring, the OH group, the disubstituted phenyl group, and then perhaps on the last ring or this tail, you can then make scaffolds that will lower log-D value. So in that case, you have the bits that will make the in vitro good, but then you also have this tail here that will make this further stabilizing effect. Plus, it also lower log-D value, which then hopefully will give you a high in vivo. So that's like where the angle that I'm going. But we don't really have any compounds like that already tested. It's just a suggestion. And that's kind of like my thoughts after doing the SAR analysis. So you can really do this tail exploration in a future experiment study, someone else's PhD perhaps, that will test like have that doubling effect of stabilizing the interaction with the access channel to the active site, and also lower the log-D value to increase your in vivo as well. That's some of my discussion.
Just quickly on the structure in the middle, the L1 linker there that you've got in the middle. In the case of that being nitrogen atom, then what you said about the OH being an unstable group makes a lot of sense. In the case of if the L1 is a carbonyl, so you have that amide functionality as the linker, you could in theory have an OH on that center, right? But we don't have anything that looks like that. Yeah.
No, we don't have any yet.
Okay, but as a synthetic target, if the OH is seen to be helpful, then that's a synthetic target that's possible.
Yeah, or we can perhaps also try like this here as well, because then the amines on the other side, and you have a carbon here, so that might also help as well.
Yeah.
Because
that's no good, is it? Right.
I actually explained this, so it's actually not no good. I explained it a bit there. There's an asterisk to that as well, which is that we did actually make an of these. So we borrowed them from Epichem, and all of the one that has this here, the core is actually an amine and not a carbon. So it's not an orange to orange comparison. So you can't really say it for sure.
Right.
But yeah, so it's both lacking the OH, and it's also changing a tertiary carbon to a tertiary amine in the middle. So this here is actually an amine. So
right.
I did explain that in the written bit. But that would probably be one of the options if we want to try that in the future.
Are you assembling a list of things that could be worth making while you're going through this analysis?
Yes, I haven't put it down in concrete terms, but yes, I do have a few.
Right, to capture the thoughts of what will be good to make next based on what pops out of this analysis.
And obviously, if Dimitri can give it the match pair for the other bits as well, I can synthesize something like this, but perhaps we do actually need to cut it down. I'm actually going to send this to Kimberly, Alice, and Peter when they're free for a little bit of a constructive feedback as well.
All right, so from your perspective, that's the next bit.
Yes. Yeah, so that's all from me.
I can go next then.
Yep, sure.
Can you see my screen?
Yes, we
Should we
can.
go to- Yeah, good. Okay, so coming back to the structure activity analysis. So I did structure motive B and motive C. So both of them are finished now. So we can kind of like wrap this up. So this is those two are like the final versions, essentially. The only thing that we need to decide is on whether this will need like further changing, because I can see that the style that you're pursuing like for the paper is slightly different like to what I proposed like on the on GitHub previously, but we can adjust to this. So essentially I was looking at the piperazine. So essentially the 2,4 fluoro-chloro substitution and pyridine ring scaffold with either piperazine or piperidine ring on the other side. Not many, I would say, we have potent compounds. A lot of them, like for example, azidine, were promising like at the beginning of the in vitro stage, but they didn't pass the 25 micromolar threshold. So either that has to be classified as an active or like medium potency, which is not very helpful as well, because it's like not giving the enough inhibition to proceed further. So motive. So motive C we discussed last time. So motive B is the piperazine, and the most potent are the carboxylates, essentially. The third butyl, the methyl group, and ethyl carboxylate. And bringing on from this point, I had on top of what has been done like for series 1 phenols, I had like a few more suggestions based on new molecules, based on the previous molecules that we did. Like, so I thought like a few more substrates I would do before sending the large batch to Wendy, including the Keroxime series. So if you look at the motive B, for example, the very first compound that was used as a positive control with the ethyl carboxylate moiety with the piperazine. So I checked the database and found that actually we don't have this molecule essentially with this substrate. But if you look at it carefully, so essentially it combines two positive moieties, like one from motive C and the other from motive B structure activity analysis. So it's the piperidine ring and the ethyl carboxylate. So essentially it's the same as the very first positive control that gave like potent results, but only with the, instead of piperazine ring, would have like the piperidine. And at the same time, it would combine the two actives. So it would be interesting to see what sort of result we would get like with that one. That's the first suggestion. Also, the previous assay back in summer 2021 gave very good results with the pyridine ring. So had a bunch of those extensions being investigated like in synthetic analysis as well. So one of the thoughts was of making pyridine with the following hydrocarbon substrate as well. And finally, I was also thinking, and I suggested this to Matt like back at the end of last year, this compound with dioxylene ring. Just a quick reminder, the dioxylene ring is a active moiety within most of the currently used antifungals against mycetoma. And I'm primarily talking about the itraconazole. The importance of dioxylene ring is that it takes part in the so-called dioxylene ring scission process. So it's essentially involved in metabolism and creates a number of metabolites. So essentially it goes first past metabolism in the organism. It interacts with this cytochrome P4/3A enzyme, but it also creates 3 additional metabolites that take part in the actual interaction with the target. So I was thinking, so I'm not entirely sure. I wanted like people just to say their opinion on this. So I was thinking at first, should we actually explore this substrate on its own? Because it's a big difference, of course, talking about like itraconazole, where we have like a specific mechanism of action, and when it creates like those metabolites, the mechanism is actually known, or just like looking at this like on its own. I think we should, because this is like slightly different, like to all what we have like in the entire mycetoma library, and because we still use itraconazole like as the current treatment option, it maybe would be useful as well. So any thoughts on this, Matt? We haven't discussed this like in much more detail, so I did like some reading on this. What do you think about the dioxylane substrate?
So with regards that itraconazole is still currently the drug used to treat mycetoma, the reason for that is that simply we don't have anything better. But if you look at treatment outcome with itraconazole, you still need surgery. And the fungus, when you take it out after 6 months of treatment, it still happily grows. So I know we explored the CYP51 extensively also with the Fennery moles, but maybe it's a better thing to go to look towards different targets as well than only CYP51, as to see how we can improve on the activity of CYP51 on its own, because in vitro it works very well, but in vivo it doesn't do much.
But do we know how it will react like with regards to our scaffold that we're using? Because it's like totally different, like to what we have, to what they have like with itraconazole. So the reason why I emphasize the fact that like dioxylane ring might be important is because it's like used in many, it's actually inserted like in many antifungals. So it's kind of like an important moiety as well. So my rationale, I was thinking like, it actually kind of like expands like and like deviates like from what Han just told us, like based on the SIR that like we did. It's just like thoughts like for the future for the series. So I thought like, what would happen like if we explore this like in the context of this scaffold, like with the pyridine ring, with this aromatic ring, like with the fluoro and chloro substitution as well. So that was my thought.
Yeah, my only worries is that I'm not because also if you look at the activities of even the best Fennery moles we created so far, maybe CYP51 is not the perfect target. Maybe we should also explore a little bit in series where we might have a different target and then combine. That might also be an option. That maybe the conclusion can be, okay, we explored quite a lot of CYP51 and this is the best we can get for now. But what if we hit the fungus at 2 different sites simultaneously? Maybe we get better activity there, because yeah, it's still a little bit disappointing, of course, that we don't get a very high survival rate in the larvae. So maybe we're looking at the wrong targets as well. So maybe we should combine and look more into combination therapy. That's the only worry I have, because at some point we need to make a decision. Are we going to explore CYP51, the Fennery moles, even further? Or are we just going to say, maybe we should look at a different series, a different enzyme to target, and see if we are successful there, and if we are successful, maybe we can combine the two. Yeah, that's a little bit what I was thinking of. At some point, we need to make this decision. And my gut feeling is that maybe we should abandon CYP51 as a target for now, and maybe come back later if we have a different target as well, in just to see if we can combine. But that's my gut
No,
feeling.
that's the reason why I'm asking it. So my strategy was like, because I'm still just about to send the large batch with the compounds that I made like last year, along with the Keroxime series, and I was like making several additions at maximum. So we're talking about like probably maybe like, so 53/1 actually has been synthesized. I just need to purify it. But it's
yes,
actually,
and of course, all the things you already synthesized, we will still screen. Don't worry about that. But at some point,
dioxazine ring has not been, the dioxazine substrate has not been added yet. So I was just pursuing like your opinion, because it's either like we go for it or not. So my understanding was like, until we test it, we don't know for sure. But you kind of seem to be like more skeptical that it's like not worth doing it. Because of the target, because of the CYP enzyme, as I understand.
I think it's worth doing because it's a single chemical step, so it's very easy to get hold of.
Yeah, that was my rationale as well, to be honest.
Yeah, so by all, I think it's, you know, worth sticking in the mix. It's not as if it's a big departure. I think synthetically, it's not like we have to make the starting material, so go for it. I think it's important to check if any compounds with that motif are available in the Epichem library.
So I checked, it's gonna be a new one. I'll double check before I do it again, just to be absolutely sure. But I was surprised to see that like the 53/1 wasn't actually done before, because essentially it's a combination of two motifs, so the green one and the piperidine ring.
But I think
which proved active like in the past.
But I think that you're, I mean, you're right, Wendy. You know, I think with this paper, you know, we want to be trying to draw a line underneath it and say, well, this is what we've done, and now we're going to look for alternatives, because there may be alternatives that are less well explored. I think in general that's a sound idea. The idea about doing some of the biology was perhaps to help draw that line, to say that, you know, we've done this and we've proven that the target is this, and here's some, you know, new structural information that we could bring. That might be quite nice. I mean, if we had a structure of the relevant protein with something bound, that would be neat. As a drawing a line out of things, particularly if there are no relevant crystal structures available so far, that would be quite nice.
Yes, and I think that the crystal structure is still valuable, because of course we're looking here at novel molecules, but we're far from the clinic yet. And with the phenerymols and maybe also with the azoles, to make a crystal structure that will help maybe in the future as well, or in the very near future. But I think that even if they are going to, if we will find a new drug, I don't think that they will treat mycetoma, if we found new drugs, as a monotherapy. I think they will, for the time being, include the CYP51 target, so the azoles, or if we get a very good phenarymol, maybe the phenarymols later on. But with fungi, in the past, it was never possible to do combination therapy, because all drugs were targeting the same pathway. They were always targeting ergosterol. So you have the azoles targeting CYP51, but you also have terbinafine, which is targeting in the same pathway. And that gave some issues with combination therapy, because there could be cross-resistance there, and there could also be upregulations of genes downstream, which actually diminished the activity of the other drug. But if we find novel targets, and now with olorafim, we already have found some new targets, the DHODH, but also in our own screenings in the libraries, the benzimidazoles we found. There are the ketoxamide seemed also important, so we might have the opportunity now to go down this road into combination therapy.
Yeah. So with regard to the paper that we have right now, is what we have so far, plus the SAR and the characterization data, are they sufficient by themselves because I'm guessing we can group the sort of like the crystal structures or those studies into the next paper, for example, with this being the stepping stone to that. So we can just get what we have right now and just finish that up into a nice package for a paper like now.
I guess it depends. Yeah, hopefully. I mean, I guess it depends how it reads, right? When it's been condensed and edited and polished, that there is a conclusion there of something that we found. If it reads like here's a bunch of compounds we've made and this is the data, it's more difficult to publish that.
Yeah,
but
because I guess we can still defend our lofty angle with the result we found so far. We bolster the evidence that it might be a CYP51. We just don't have a crystal structure yet. But the SCI essentially has never been done before for phenaromals on here. So this is the first time ever that it's been done. So we found these new patterns that is consistent with this mode of action. And our in vivo that we have here also kind of like support what we have from the previous paper as well regarding the lofty versus in vivo as well. So it's, yeah.
So just to finish up what's on the screen, Dimitri, so those three molecules you talk about, yeah, we should make them and see how they go.
Yeah. Okay. I've got like four more phenaromals to synthesize, and then I'm kind of, and one catexime that didn't work like previously, and then I'm ready to send the batch.
Okay.
To Wendy. Yeah.
Okay.
I've got to transfer to another meeting in a couple of minutes. If anyone had urgent things that they wanted to mention.
Just to wrap up, like with the catexime, so we have like 5 compounds that I proposed like previously. I've got at the moment like 4 are complete. One I changed the protocol, which did not work, so I need to synthesize that. And as soon as I do that, then and along with those 4 phenaromals, I will send the batch to Andy. So it will be surplus of around like 10 molecules, along with like 3 YMCA phenaromals. So it's gonna be like a bit of extended one. Also on the catexime series, it's always just a reminder, it's always a mixture of EZ isomers. So I found a method how we can favor the equilibrium towards the Z isomer, which is useful, especially to those with respect to those compounds that have proven to be effective. So primarily I'm talking about the peripheral exposure control, which is also a mixture of EZ isomers. And essentially, I think the next logical step would be to separate them and test them separately, just to see like how it would behave. Like, so that's like probably the future work for the series as well. But those compounds will the one that you see the substrates will be tested first. Will be the mixture of EZ isomers.
Yeah, so just a quick note on the benzimidazole series as well. So Kimberly and I, as we mentioned in our November meeting, so in the next few months we are going to make some new compounds from that series as well, just to for our future work. That will be the other targets to avoid that mono drug mechanism of action. So we're just looking at benzimidazoles as well. So we're gonna meet up and strategize for our sort of like next batch of compounds.
Okay, if you could post some of that stuff, that would be very useful, because I'm talking to the Sevenoaks School about exactly that, and they would love to take part in this. And then in a few months, I'm giving a talk to heads of chemistry, essentially, for a bunch of schools down in the southeast of England, who may also want to jump on board if there is a good protocol that the students can follow. So there may be a sudden widening of interest in this if the chemistry is doable in a school lab.
So another thing as well is that Alice, Peter and I got funding to run an outreach program with high school students at UCED this year. And we put in to have about 100 students, and we have a school with 100 students who wants to come. And we're planning on continuing the series 2 stuff, I think, because that's what we've previously done. So we can just sort of repeat. But we're also potentially looking at, because it's through the Drug Discovery Institute and they want to expand and get more people involved in research projects together. So they also want us to expand a bit and do some computational chemistry. So we're going to have a chat with some people see if we can start a collaboration there. But we'll update on that and see what we can find anyway. And also some biological stuff. So we might find that some of the drugs that we make, we might want to test them against something else, and we try and build a collaboration with that too. But the whole point is that it will be outreach, and it's supposed to all be high school kids doing it. But there's just some news on that front as well.
Great. I'm sure Ben Perry could help with some of that stuff.
Mm-hmm.
Okay, one small thing for the meeting. Matthew suggested that we make a screenshot with all of us.
Oh, yeah.
Maybe we should do that now then.
Yeah.
And who's
good.
going to host it on the social media?
Oh, well, I can post it for sure and tag everybody.
Okay, yeah. So if Dimitri can stop sharing.
Good, yeah, I'd forgotten about that. It's always creeps up on me, and this time it's on a weekend, so we're going to forget. All right. All right. Dimitri, Mickey, if you want to be on this, you have to unshare. I mean, sorry, you have to screen on.
Yeah, you have to put your camera on, Dimitri.
Yeah, I understand. It's just, I think my safety settings are not like up to date.
All right.
So I think the camera is on.
Everyone ready? Everyone ready? I guess
Yes.
so. All right. Wait a minute. Wait a minute. All right. Cheese.
Cheese.
I think that's it. Hang on a minute. I can probably try and share that. No, I'll share it. It's very civilized, that as a photo. So I'm sure that's fine. I'll send it around by email before if anyone wants to object, but that's a pretty nice photo, actually. I think all of us are looking reasonable.
Okay.
Which has never happened before on a Zoom call. Okay, great. Anything else? I guess we can just take up on the issue after the meeting. How about that?
Yes, I think that's okay because we're past the hour already. Okay, then see you all next time.
Right.
If you can
Bye.
send me that that updated match pair that you've done for B and C, I can play around with it as well and see how it goes.
Sorry, are you talking to me?
Yes.
Wait, but like it's been posted on GitHub.
Oh, is it posted up? I didn't.
Yeah, on this, on the SAR last week.
I'll look at it. Thanks.
But let me know. I'll send it to you if needs. Yeah. Goodbye, everyone.
Yeah.
Bye.